Hello, this is David Coyle, and welcome to the next video in our series on Illumina Multiplexing. In this video, we're going to go over the background and principles of barcode design. Just a quick review, the videos in this series consist of the overview, this video, which is the background and the details of barcode design, and the next video will be a tutorial on using the barcode generator. And then finally, we'll have a video that shows some applications and examples of barcoding. We should note that all of these videos assume a basic familiarity with Illumina sequencing. But just as a quick review, here is the Illumina Y adapter, and here's the barcode. This is the sequence that you're going to add to the adapters to uniquely identify a particular pool of sample. So, there are a few considerations when designing barcodes. The first is the balance between the four nucleotides at each position in the barcode. This is because the way Illumina determines clusters is based on the first few bases. And if the first few bases are the same, then Illumina can't determine what belongs to one sequence versus another. For example, if the readout from 4 different barcodes is as shown here, then the first two nucleotides of every sequence in the run will be AT, and Illumina can't determine what a cluster is. We should note, and we're going to go over this later, that this is the readout of the barcode. The actual name of the barcode will be the reverse complement of that, but we'll get to that. So one way to avoid this is if you're only mixing two libraries, you should use two barcodes for each library to improve the nucleotide balance so that each of the four bases is represented about a quarter of the time in the initial cluster determination. However, inappropriate cluster detection can still be a problem with some versions of Illumina software, particularly the newer high-seq machines, but we anticipate that this will change with time. So, other considerations when designing a barcode. The length of the barcode is important. We use 5 bases. An odd number is better than even for avoiding palindromes. Barcodes could be as short as 4 or a little longer if necessary. The difference, or the genetic distance between barcodes, is technically called the Hamming distance. We find that 2 bases is usually sufficient, but in some cases 3 bases is fine. But if you're using short barcodes and you try to make them too different, you won't have enough combinations. A couple of technical notes. One is that you need to order the adapter with a 5' phosphate on adapter A. Normally adapter A, and this is our nomenclature, is adapter A for the paired and Illumina adapter, the first one. Normally that comes with a 5' phosphate. Because we're going to add a barcode to it, we still need to remember to add a 5' phosphate upstream of that barcode. And no special purification is needed. The standard desalting is fine. So a quick note on nomenclature. We name the adapter by the way it shows up in the top sequence, what we call adapter A. However, the sequence in the read will be the reverse complement of that. So let's look down here at the Illumina paired end adapter sequences in blue. This is the standard adapter A and adapter B. Note the 5' phosphate on adapter A. When we add a barcode to this, we add 5 bases and then an A. And this A is to allow the sequencing primer to bind. There's normally a T overhang present there. And then here in adapter B, you can see that we have the reverse complement of that, and then we've added the T overhang again. And this is the equivalent of this T overhang here. So by convention, we call the barcode this, TTTCC, but in reality, all the reads in the sequence will be GGAAA, and we'll get to that in a minute. So when you want to design barcodes, you have 3 choices. The first is they can be designed by hand, and this works fine for simple experiments. Just choose an appropriately balanced series of bases. Perhaps even simpler than that, we've created a list of 24 well-tested barcodes available on our website at the URL shown here. And the most common option is to use a barcode generator, and we've made a simple Python barcode generator available on our website, shown at this tiny URL here. And in the next video, we're going to go through how to use this barcode generator. And this takes into account several parameters: the length, number, genetic distance, etc. Something important to note when dealing with barcodes is the critical importance of measurement. If you think about a sample that has 96 different barcodes in it, that means that any given barcoded sample only represents about 1% of the total sequence in that run. So if you want to be able to compare between samples, you need highly accurate measurement. And so there's a couple of pitfalls that we've encountered that we wanted to share with you. One is that a nanodrop spectrophotometer measures free nucleotide as well as DNA. So if you're going to use a nanodrop, be sure to purify your samples before using the nanodrop. Another is that RT-PCR, which is commonly used, can amplify some sequences more than others, so it's also risky. We recommend the use of double-stranded DNA-specific fluorophores. For example, we use cybergreen. This is a simple, replicable, plate-friendly method for measuring the concentration of DNA in your libraries. So let's go over the barcoding workflow real quick. You design the barcodes using one of those three methods, order the new adapters, being sure to get a 5' phosphate in the right place, do your standard library prep and quantification, preferably using cybergreen or some equivalent, pull your samples and submit them for sequencing. And then we get to data analysis. We'd like to say a quick word about that here. So when you're dealing with barcoded sequence, you need to first process the sequence file to remove the barcodes. The first thing to do is verify that the barcode is intact. And again, we already went over this. Make sure that you are looking for the reverse complement of the name of the barcode. So in the case of TTTCC, you're looking for GGGAA on all sequences, even those that came from adapter B, because of the way the Illumina primers line up. Then you're going to remove the barcode from the read, paste it in the name, and don't forget to remove the extra T as well, and the 6 matching characters from the quality notation line. We'll show you what that looks like here. Here's an actual Illumina read. You can see I've highlighted in blue the barcode and then the T overhang. So for processing, we simply cut this, add it to the sequence ID, remove the T, and then cut out those 6 characters of notation, and now you have a process sequence, and now you can sort into different files or whatever it is that you want to do now that you've removed the barcode from the sequence. So that's it for this video. In the next video, we're going to go over how to use the barcode generator. Thanks for watching, and see you then.